Greetings, Brookings Bio students. This is Mrs. Rydell, and today we're going to look at the last part of the pathway for cellular respiration. We looked at starting with glucose and using oxygen to burn our glucose, break it down by going through the steps of cellular respiration. We talked about glycolysis, splitting our sweet to make pyruvic acids. In our last video, we took those pyruvic acids and we fed them into the Krebs cycle. We made some ATP, we made some carbon dioxide from the carbons that started in glucose, and we charged up a whole lot of those high energy electron carriers, some NADH and some FADH2, and we're going to look at where those electron carriers go next, and that's into electron transport. So if we look at where we started, glycolysis was out in the cytoplasm, outside the mitochondria. We're feeding in glucose now, and we made a little ATP in that process. Then we made split our sweet with glycolysis, made pyruvic acids that we fed into the mitochondria, into the matrix of the mitochondria for the Krebs cycle. And now we're going to break apart those carbons a little at a time, and we made a little ATP there. But a whole lot of these high energy electron carriers, the NADH and FADH2. And if you remember back in glycolysis, we made a couple NADH carriers back in glycolysis when we split our sweet apart and made pyruvic acid. Those also can feed into the mitochondria and become part of the electron transport chain. That's our next step in the pathway here, electron transport, ET, and we're going to get an awful lot of ATP out of this third part of the pathway. And the enzymes to do the electron transport, remember in photosynthesis we attached our electron transporters to a membrane so they could pass in line in sequence. And the closest membrane to where Krebs cycle is happening, we're making these electron carriers, is the cristae membrane. And so our electron transport chain molecules that are part of that transport chain are going to be attached. They're part of that inner mitochondrial membrane, part of the cristae. So we're going to watch a little movie about our next part of the pathway, which is electron transport.
Most of the energy a cell harvests during the Krebs cycle is held in energy carriers such as NADH and FADH2. These energy carriers stay within the matrix of the mitochondria. They transfer their high-energy electrons to proteins on the inner membrane of the mitochondria. The inner mitochondrial membrane separates the matrix from the outer compartment. Large proteins are embedded in the inner membrane. These proteins are the electron carriers in the electron transport chain. The electron carriers are arrayed side by side, like the links in a chain. 3 of these proteins are ion pumps. NADH transfers 2 high-energy electrons to the first electron carrier, an ion pump. NADH changes to NAD. The carrier protein uses energy from the electrons to pump 2 hydrogen ions from the matrix into the outer compartment. The electrons are then passed to the next protein in the chain, and then on to the third electron carrier. This third protein is also an ion pump. It uses a bit more of the electron's energy to pump 2 hydrogen ions through the membrane. The electrons shift again to the fourth protein, and then on to the fifth protein. This protein is a third ion pump. Using energy from the electrons, the protein pumps 2 more hydrogen ions into the outer compartment. The 2 electrons are now much lower in energy. They combine with 2 hydrogen ions and an oxygen atom. Oxygen, absorbed from the air when you inhale, is the final electron acceptor at the end of the electron transport chain. A molecule of water, H2O, is formed. A similar sequence of events takes place with FADH2. FADH2 transfers electrons to the second ion pump in the chain. The FADH2 is converted to FAD. hydrogen ions are pumped through the protein into the outer compartment. The electrons shift down to the last protein in the chain. More hydrogen ions are pumped. Again, oxygen serves as the final electron acceptor, and a water molecule is formed. Ion pumps are powered by energy from electrons. The ion pumps move hydrogen ions from the matrix into the outer compartment. Hydrogen ions concentrated in the outer compartment represent a form of potential energy, like water held behind a dam. They diffuse back into the mitochondrial matrix through a special protein called ATP synthase. This diffusion of hydrogen ions through the ATP synthase energizes the synthase. This energy is transferred to an ADP molecule forming ATP. This formation of ATP provides the cell with large amounts of usable fuel.
Okay, so we're going to send our electron carriers up to the cristae membrane. This is our cristae here. You can see the phospholipids, heads with the tails. And remember, it separates the compartments. We have on this side our intermembrane space, the space between the cristae and the outside membrane. And on this side is going to be our matrix. And remember, these carriers are being made in the matrix as part of that Krebs cycle pathway. And so all they have to do is float over to the nearest cristae membrane to drop off their electrons. And these electron transporters now, they're kind of set up in the membrane just like they were for photosynthesis. We have the electron transport chain with ATP synthase at the end. What's missing here, though, that we had in photosynthesis? What's missing is those photosystems. Remember, in photosynthesis, what was supplying electrons to our electron transporters were the photosystems being energized and passing electrons down the line. Now we don't have those photosystems. We're in the mitochondria now. This is not in an autotroph. This is a heterotroph. But electron transport is going to work the same way. We're going to feed in electrons. Now they're coming from these electron carriers, the FADH2 and NADH, instead of from the photosystems. But we're going to pass the electrons down the line. We're going to use those passing electrons to pump hydrogens into our space here. Before we pumped hydrogens into our thylakoid space. Now they're going into the intermembrane space. And we're going to build up a potential behind this membrane here with all the hydrogens. And then they're going to want to go from high to low. And remember, ions don't like passing through membranes, but if we give them a tunnel to pass through, an ion channel, they can get back through the membrane. And remember, ATP synthase is our ion channel to allow those hydrogens back through the membrane, and the power of the hydrogens moving through ATP synthase are going to be used to charge up our ADP into ATP, and our carriers now are going to supply the electrons to make that happen. Notice they drop off in a little different place. The NADH drops its electrons on the very first carrier, and these are, remember, proton pumps. Every time they pass electrons, they're using the power of electrons to pump hydrogens. 1 pump, 2 pumps, 3 pumps. When NADH drops its electrons and goes back to being NAD+, the hydrogens get pumped 1, 2, 3 times into my intermembrane space. And that means 3 ATPs coming back the other way. FADH2 drops its electrons here. It's a little farther down the electron transport chain. It's going to skip that first pump. So when FADH2 drops its electrons and goes back to being FAD, it's only going to hit 1, 2 of the pumps. It's only going to pump hydrogens twice, and that means 2 ATPs coming back when it comes back through the tunnel. So electron transport. What do we need to remember about this? High energy electrons are going to be coming from our electron carriers, our NADH and our FADH2. They're going to be passed along the electron transport chain from one to the next, and the energy from moving those electrons are going to be used to pump hydrogen ions across the membrane into our intermembrane space. The pumping of hydrogen ions into that intermembrane space represents potential energy. It's like water behind a dam. And we can use that power as the hydrogen ions come back through ATP synthase through the ion channels back to the matrix side, we're going to use that power to stick the phosphate groups onto ADP to make ATP. Oxygen is going to serve as our final electron acceptor at the end of the electron transport chain, and combination of oxygen with 2 electrons and hydrogens is going to make a water molecule. Let's take a little look now at those electron carriers and the amount of ATP that they're going to produce, because remember, they're dropping off in 2 different places. Our NADH carrier is going to drop off right at the top of the electron transport chain. So it's going to pump 3 times. It's going to hit 3 of those proton pumps. But the electrons from FADH2 are going to drop a little bit farther down on the transport chain. So they're going to skip that first pump and only pass electrons through these 2 pumps. So we're only going to pump 2 times. And that means those 2 electron carriers are going to make different amounts of ATP. 3 pumps means 3 hydrogens into our space, and that means 3 ATPs when we come back. FADH2, even though if you look here, the H2 kind of thinks like that's got more hydrogens, you'd think it would make more pumps and make more ATP because it's got more hydrogens. But what's passing is the electrons, and the electrons are creating the gradient. So FADH2, because it skips that first pump when it drops off its electrons, we're only going to get 2 ATPs back. So let's take a peek at the big picture now. Remember, in glycolysis, out in the cytoplasm, we're going to make 2 ATPs out of that process. We're going to split our suite and get 2 pyruvic acids that are going to feed into the Krebs cycle. And Krebs cycle in the matrix of our mitochondria now. We're going to make 2 ATPs there, 1 for each pass as it goes through, and charge up a whole lot of those electron carriers. And those electron carriers now, the NADH and FADH2 we made in the Krebs cycle, are going to feed into that electron transport chain, as well as those NADHs that we made out in glycolysis. Those can also be fed into the mitochondria and pass their electrons to the electron transport chain and make a lot more ATP. Remember, every NADH that drops off is making 3 ATPs, and for every FADH2 that drops off, we're making 2 ATPs. So we're going to get a lot more power now out of this final phase. So let's kind of do a little summary here. First, we're starting with glycolysis, and respiration is made out of these 3 steps now: glycolysis, the Krebs cycle, and then finally the electron transport chain. And remember, this is our with oxygen pathway. We're burning our glucose with oxygen, and we're going to get a lot more power compared to doing fermentation. Glycolysis happens in the cytoplasm, outside the mitochondria. We're going to break down glucose, 1 glucose molecule, into 2 pyruvic acids or pyruvates. And remember, we put in 2 to get it started, but got 4 back for a net gain of 2 ATPs and 2 NADHs. We're going to feed now, with oxygen available, feed our pyruvic acids into the Krebs cycle. We're going to take those pyruvic acids in the matrix of our mitochondria and break them down, take the carbons off one at a time. And for the 2 pyruvic acids, I'm going to get 2 ATPs, 6 carbon dioxides. Remember, glucose had 6 carbons in it, so 6 carbon dioxides. I'm going to get 8 NADHs and 2 FADH2s. Remember, for the test, we were not worrying about the intermediates, but you should have a feeling for what's being produced, where the carbons are going, what kind of power molecules we're making, what's being produced during the Krebs cycle. And then finally, we're going to take those electron carriers that we made in Krebs and feed them into the electron transport chain. And the enzymes for doing electron transport are part of the cristae, that inner membrane of our mitochondria. We're going to use high energy electrons and the hydrogen ions that are donated from those electron carriers, and we're going to make ATP, 32 net ATPs. So oxygen is going to act as our final acceptor to produce a water molecule, and that finishes out our process. Remember when we started with photosynthesis, we were using carbon dioxide and water. Water was donating the electrons to chlorophyll as they were passed down the line, and I split water to replace those. And we were making glucose. And remember we said the reverse cellular respiration is the reverse of photosynthesis. We're going to take our glucose, and our final molecule that we end up with is our starting molecule that we started with in photosynthesis, and that's a water molecule. Okay, so kind of compare the phases here, because for one glucose going through all the way through Krebs cycle and into electron transport, if you look at our comparison, for glycolysis we only made 2 net ATPs. For Krebs cycle, counting the charging into acetyl-CoA and feeding it into Krebs, we only got 2 ATPs there directly. And then for the electron transport now, look at how many ATPs we're going to get out of our total, for a total of 36 ATPs, and most of those are coming from those electron carriers that we made during the electron transport chain. So cellular respiration, aerobic with oxygen, one glucose is going to net us 36 ATPs. Compared to fermentation, remember with fermentation without oxygen, one glucose only got us 2 ATPs. So doing respiration with oxygen is a way better way to make power molecules than running fermentation. We get way more ATP burning our sugar with oxygen than we do with fermentation. So let's kind of make a connection here between how this works in our body, because we're doing this to get the power. And remember, carbohydrates are power molecules. They supply energy to our body. When we talked about carbs back in our molecule chapter, way back at the beginning of the year, we said carbohydrates are fuel molecules. Your cells burn glucose for energy. If your body was a car, glucose is the gasoline that makes the car go. And we're burning glucose molecules now as part of that process of getting energy. So when you exercise short term, cells normally only contain a small amount of ATP. And we use those ATP molecules. They get charged up by doing glycolysis in the respiration pathway. But really, we only have a few seconds of ATP laying around in our body. And once that ATP gets used up, then we can recharge it again. Lactic acid can give us power for about 90 seconds. So short term, remember when we first start running, think if you're sprinting down the hall because you're late to class, you start running and you're kind of panting a little bit, maybe a little low on oxygen right at the beginning. The cell is going to be, for short, quick bursts, maybe just a minute and a half, we're going to be doing lactic acid fermentation. Then once the race is over, that lactic acid can be broken down using oxygen. You stop running, you get your breath back. And well-trained athletes actually burn their lactic acid more efficiently. That's why you train. You activate those enzymes. You're easier to recover from the lactic acid that builds up in your muscles. If our athlete, though, is going to be running longer than just a couple minutes, the only way that they're going to get power enough to keep running, to run and do the things that they need to do, is through cellular respiration. It's the only way to make enough ATP. Remember, fermentation doesn't make any ATP, and the glycolysis has only given us a couple ATP. So if we want to run longer, if we've got a soccer game where you're running for 45 minutes, or you're running back and forth on the basketball court, or doing a marathon, cellular respiration is the only way that you're going to get enough energy that you need. And remember, respiration is releasing that energy slowly over, burning our sugar, releasing a little at a time, the power, and charging up our ATPs. And That's why athletes condition themselves. So what happens in a long race when your glucose gets used up? You're burning, remember when we eat food, it puts glucose into our bloodstream, and you have a little bit of sugar there for energy needs if you do a quick run or you're starting to play a sport. But remember, we have backup in our glycogen bank. Animals store glycogen by hooking glucose together, and they save it for later. You save it in your liver, you save it in your muscles. If you're an athlete and you carb load before a big game, or you have a tournament, you know that you eat carbs the night before. You load up on pasta or pancakes, spaghetti, and it's because you're loading your sugar bank. You're taking that excess sugar that you're eating and saving it for later the next day when you're running. And so when you're an athlete and you use up the glucose that's in your bloodstream, you're going to switch over to burning glycogen. And all you have to do is lop off those little sugar molecules one at a time, and you get glucose to burn for cellular respiration. Muscle cells store glucose as glycogen, and that can be broken down when the cell is doing respiration, when it needs. Probably you have enough glycogen stored in your muscles as backup for about 15-20 minutes. And then after that, we need something else to burn, and our big backup is fat. Fat is the way your body stores energy for long term. And that's why when you do exercise, if you're trying to exercise to lose weight, aerobic exercise, you have to go for longer than 20 minutes before you break into your fat reserves, because those first 15-20 minutes you're burning the glucose in your bloodstream, you're burning your glycogen in your glycogen bank, and you really don't start burning fat until you get to after about 15-20 minutes of aerobic exercise. Let's talk a little bit about different kinds of organisms, because remember, eukaryotes, plants and animals, fungi, protists, have mitochondria for doing their cellular respiration. But prokaryotes, remember, they don't have membrane-bound organelles. They don't have any mitochondria. So how can they do respiration? They attach their electron transporters to the only membrane that they have, and that's their outside cell membrane, their plasma membrane. And they can still do respiration, they just need to make a little compartment. So here's how this works. I'm going to draw a little picture here. If this is my bacteria cell, I'm going to make a little cell. And what the cell does is kind of make a little pooch out place. Outside the cell. And remember, bacteria have a little single loop of DNA. I'm going to put a little DNA loop in here. But they don't have any organelles now. No mitochondria, no endoplasmic reticulum. So what do? They pump their hydrogens out of the cell, actually. They make a little, kind of a little indent, a little like a little bay. Put the hydrogens out of the cell in that little area, and then make a little concentration gradient, just like we do in our intermembrane space. And then once I get the hydrogens concentrated out there, I can run them back through ATP synthase to get them back into my cell. So bacteria cells can do cellular respiration also. They just don't do it by putting their hydrogens in a compartment inside mitochondria. They put their electron transporters in their cell membranes and actually pump the hydrogens out of the cell and then bring them back in again. That's the end of our respiration video. We've talked about all 3 steps: glycolysis, Krebs cycle, and electron transport. And thanks for watching.